Hey guys! In this video, we'll be going over an introduction to bioinformatics and creating our first function, pattern count, with a Python coding language. Before we begin, let's start off by understanding what exactly is bioinformatics. Bioinformatics is the science of collecting and analyzing complex biological data such as genetic codes. It's the intersection of both computer science and biology to help solve problems that research scientists face every day in the lab, using efficient and usable algorithms to get to a solution. Computational methods are now the only realistic way to answer many questions in modern biology. First, these methods are much faster than experimental approaches. And second, the results of many experiments cannot be interpreted without computational analysis. So what exactly are these problems? Well, the first one that we will be looking at is genome replication. Genome replication is one of the most important tasks carried out in the cell. Before a cell can divide, it must first replicate its genome so that each of the two daughter cells inherits its own copy. The replication process begins with a pair of complementary strands of DNA and ends with two pairs of complementary strands. Replication begins in a genomic region called the replication origin, denoted ORI, and is performed by molecular copy machines called DNA polymerases. Locating the origin replication presents an important task, not only for understanding how cells replicate, but also for various biomedical problems. For example, some gene therapy methods use genetically engineered mini genomes, which are called viral vectors, because they're able to penetrate cell walls, just like real viruses. Viral vectors carrying artificial genes have been widely used in agriculture, and was first used successfully on humans in 1990 for immunodeficiency disorder. The idea of gene therapy is to intentionally infect a patient who lacks the crucial gene with a viral vector containing an artificial gene that encodes a therapeutic protein. Once inside the cell, the vector replicates and eventually produces many molecules of the therapeutic protein, which in turn treats the patient's disease. To ensure that the vector actually replicates inside the cell, biologists must know where the origin of replication is in the vector's genome and ensure the genetic manipulations that they perform do not affect it. Biologists would usually plan an experiment to locate the origin replication, for example, deleting various short segments from the genome and eventually finding a segment whose deletion stops replication. However, such existing experimental approaches to the origin replication prediction are rather time-consuming. As a result, the origin replication has only been experimentally located in a handful of species. Thus, we would like to design a computational approach to find the origin replication to save time and money. We will focus on the case of finding the origin replication in bacterial genomes, most of which consist of a single circular chromosome. Research has shown that the region of the bacterial genome encoding the origin replication is typically a few hundred nucleotides long. We will begin with a bacterium in which the origin replication is known, and then determine what makes the genomic region special to design a computational approach for finding the origin of replication in other bacteria. We will be using the chromosome of Vibrio cholerae, the bacterial cell that causes the disease cholera. The initiation of replication is mediated by DNAa, a protein that binds to a short segment within the origin of replication known as a DNA box. You can think of the DNA box as a message within the DNA sequence, telling DNAa, bind here. The importance of the DNA box within DNA replication means that it is likely for it to occur very often in the DNA. Operating under the assumption that DNA is a language of its own, let's see if we can find any surprisingly frequent phrases within the origin replication of Vibrio cholerae, which will later lead us to finding where the DNA box is and finding the origin replication in other genomes. For our very first algorithm, we will begin by counting how many times a certain pattern shows up in a string version of Vibrio cholerae's origin of replication. Note that we will be using the Python coding language to code all our projects. To begin, we will put all our code in a function called pattern count. We define this function by writing def pattern count, open close parentheses, and then colon. A function takes in an input and returns an output. Placing code into a function is useful because we can call later this function by name anytime we want, instead of having to repeat all of the lines of code contained within the function. The keyword def at the beginning of a function signals that we are defining a function. Inside the parentheses will go the parameters for our code, which will be text and pattern. We will then define a variable called count and set it equal to 0. Next, we will define our for loop. For I in range len of text minus len of pattern plus 1, and then colon. Len is a function that counts the length of a string. Here we use len to return the length of text and pattern. The keyword range allows us to start counting from 0 to a certain value minus 1. In our code, we set range to start counting from 0 all the way to the length of text minus the length of the pattern. Note that we had to add 1 since range runs from 0 to the value minus 1. This for loop allows us to iterate through the string of text. Next we will write if text I colon I plus len of pattern equals pattern, then count equals count plus 1. This if statement allows us to look from position I of text to the length of the pattern plus I, and checks if that part of the text is equal to the pattern. If it is, then count increases by 1. If not, then we exit out of the if statement and go back to the for loop, increasing the value of I by 1 automatically. Finally, we will write return count. This allows us to return the value of how many times pattern shows up in the text. Note that in Python, it's important to indent properly. The text editor where you're writing your code will usually help you with that, but by being aware of how you indent your code, you will save yourself time later by not having to go back and see where the indentation was wrong. Here's an example of how the code works. In this example, the text has a length of 13, and the pattern is 3 bases long, which we call as a threemer. Here the pattern we're looking for is ATA. With our code, the for loop goes through the entire text, stopping at position 11, which is equivalent to the length of the text minus the length of the pattern plus 1, as we wrote in the for loop. These blocks, or windows, allow us to see the patterns within text, and is similar to how the if statement works by allowing us to see each section of the text from I to the length of the pattern plus I. Then we compare the window to the pattern and see if they are the same. If it is, then count increases by 1. If it's not, then we go to the next window. In this example, the pattern ATA shows up 3 times in this text. Now let's apply this code to the Vibrio cholerae origin or replication. Since we already defined our function, we now need to define the values for text and pattern. We will set pattern equal to TGA TCA, and then text equal to Vibrio cholerae's origin or replication, which is this. The full Vibrio cholerae origin replication will be on the same page as this video. Finally, we will write print pattern count text, pattern to print out the result of calling pattern count with this pattern and text. On the console, we will see the value 8 printed out, meaning that TGA TCA occurs in the Vibrio cholerae origin replication 8 times. I hope this video helped you to understand what bioinformatics is, decode the pattern counts function, and how to apply it to the Vibrio cholerae origin of replication.